This is a DNA molecule, and in a certain region, there is what we call a gene for a protein P53. And in this gene, there are specific nucleotides. Now, if the nucleotide order is changed, one of the things that could happen then is that the structure of the protein that is supposed to be encoded by this gene would change. So for example, in this example, if the normal amino acid proline is replaced by threonine, then that could result in a protein that doesn't work, and as a result causes cancer. And we could go through then, endless other examples where if you change the order of the amino acids in a protein, if this one is replaced by this one, now the protein doesn't work. And many of the genetic diseases which we are concerned with involve then proteins that aren't built properly, where the right amino acids are not put in place. Each amino acid has its own chemical characteristics, and if you don't put the right amino acid in the right place, then that could mean a protein doesn't work. And if this is an essential amino acid, it could mean that someone could then be missing something which is essential and might not even be born alive. If this is that essential that many miscarriages simply result from an essential protein not being able to be made. Well, how are these proteins constructed? Well, previous videos have gone through how the DNA message can be copied in the process of transcription to make a messenger RNA. And that when this messenger RNA then reaches the cytoplasm, its triplet code then tells the cell which amino acids should be put in which order. So this video here on translation is actually part of a sequence of videos which go through not only DNA, which is on chromosomes, but which then gets copied into the mRNA in the process of transcription, and then using the tRNAs which were discussed in the previous video in this sequence, then translation can occur. And so this lecture is then on how does the DNA code then get converted into a specific order of these amino acids, what we would then call a polypeptide or a protein. In the previous video, one of the key components was mentioned, the tRNAs, the transfer RNAs. So while the messenger RNA is going to get converted into a protein, the messenger RNA is not the final product. It is a code for a protein that is the final product. There are RNA molecules where they will not be ultimately converted into a protein. The RNA is the final product. So tRNAs are an example of this, and as mentioned before, tRNAs have two important ends. One end which binds an amino acid, and then one region where three nucleotides form an anticodon, which matches the codon of the mRNA. That will be an important topic in translation. Also, however, there is another form of RNA which needs to be mentioned, which does not get converted into protein. It is what is known as ribosomal RNA, and this then forms one of the principal parts of cells, the ribosome, and as I can mention in the next lecture, probably one of the basic components in the ancestor of modern cells, the ribosome. So ribosomes are huge structures. And these huge structures are where proteins are made. And since to a large degree, proteins run our bodies. This is then an essential component for life. So ribosomes, and when we ask what are ribosomes made of, first of they are made of two subunits. So there's a large subunit and a small subunit in both prokaryotes, the bacteria, and eukaryotes. And if you ask what is this made of, one of the components are molecules of ribosomal RNA. So DNA can get copied into RNA. And while the RNA which we're discussing today is that which is then being translated into a protein, many RNAs do not get translated into proteins. And so this RNA is the final product. This RNA is the final product, as is this one. And so ribosomes have large molecules of ribosomal RNA, which are the end products of their genes. There are DNA sequences which code for ribosomal RNAs. They are transcribed into ribosomal RNAs, but never become protein. The S here stands for Spedberg units, and this is just a connotation of size, but it's not size that we can add. So don't attempt to add these units to the 5 and the 23 to come up with a total in that sense. So there are pieces of RNA, and then also proteins, lots of them. And so here's a fascinating aspect of these ribosomes, which can float freely in the cells or be part in eukaryotes of a membrane system known as the rough endoplasmic reticulum. It is the presence of ribosomes that makes the rough endoplasmic reticulum rough in its appearance. There are then a number of small proteins which then associate with this ribosomal RNA. So this is fascinating for so many reasons. First, it's this huge complex which involves, so the small unit is not just formed by this 16S piece of RNA, but also then 21 separate proteins. And here then we have 34 separate proteins part of the large subunit in ribosomes. And then as you can see, even more in eukaryotes. And so it's quite a complex structure with huge pieces of RNA and lots of little proteins involved. Later, we can discuss aspects of the origin of life. One of the models is that RNA used to be the primary molecule of cells, and it is thought that proteins came into the mix, not because they were initially the doers of the cell, like say enzymes, which are proteins which make chemical reactions happen today, but rather because they could help RNA maintain a three-dimensional shape. So that's one of the advantages that proteins have over RNA molecules. They can hold a three-dimensional shape better, and it's thought that for a while, there might have been these complexes of RNA, which was, say, doing the work, and then small proteins, which were helping to maintain the RNA. Another example of this would be the spliceosome. The spliceosome edits the genome, but it has pieces of RNA, small nuclear RNA, snRNA, and then also lots of proteins as well. And so these were thought to be kind of an intermediary between the RNA world and a protein-dominated metabolism. So here, these ribosomes are incredibly complex. The fact that the prokaryotes and eukaryotes share so many features suggests in the last common ancestor of living things, much of this machinery was already in place. But the fact that there are differences not only implies that differences have evolved over time, but that maybe translation wasn't perfected by the last ancestor of all lineages, that there were a number of modifications. We'll notice the difference between, say, translation in prokaryotes and eukaryotes in a couple of times in this lecture. So the ribosome is shared among all cells. This is where cells make proteins. But there are differences. So in this animation here, there is a small subunit, a large subunit. This is the mRNA, which was copied from the nuclear DNA, or at least the DNA. And then here's a chain of amino acids being made, forming a protein. So prokaryotes and eukaryotes make their proteins this way. All cellular life does, but there are differences in the number of proteins and the exact identity of them, and the sizes of the molecules of our RNA. Okay, and so I go through some of them here, and we'll see these again later. And so it is a complex structure of the ribosome. So here are the pieces: the messenger RNA, which was produced during transcription; the transfer RNA, which has bound to an amino acid; and now the ribosome. Now, although I won't always depict these in the upcoming videos, there are more proteins involved, some of which are known as initiation factors. And so the messenger RNA has a GTP cap. If you recall, that's a guanine, which is more or less upside down, a 5' to 5' bond, which shows where the mRNA starts. The ribosome comes in these two pieces, and then the tRNAs bind the amino acids. These are the three starting components, but there are proteins which are helping each of the three. So there are initiation factors which bind the small subunit of the ribosome, bind the GTP cap, and bind the the tRNA, which has bound methionine. So while we often just depict a translation without these, there are more components which are involved. The small subunit binds the GTP cap. Once again, that's where the ribosome knows that this is where a message is starting, and it will then slide along the messenger RNA until it reaches the first triplet, which reads AUG, adenine, uracil, guanine. That AUG codon will then be recognized as the start codon of translation. This first tRNA has an anticodon UAC, which matches that codon. Once we have the tRNA, the small subunit of ribosome, here at the start codon, and then the large subunit of the ribosome can bind, and a new tRNA can bind to the A site. Let me repeat that. So once again, there are proteins called initiation factors, which are interacting with the ribosome, the tRNAs, the messenger RNA, etc. So they vary. Eukaryotes have more, 11, as opposed to the 3 in prokaryotes. And so you can see that this is complex, and in eukaryotes more complex than in prokaryotes. And so one binds the GTP cap, that's the guanine, which is bound to a 5' bond. One binds to the tRNA, which has the anticodon UAC, and then the small subunit of the ribosome. Now, once again, there are some differences between prokaryotes and eukaryotes. One is in the existence of a SHINE-DELGARNO sequence in prokaryotes. So the sequence AGG is important in translation in prokaryotes. Mutations here can block translation of a gene. Eukaryotes do not have this sequence. So once again, while the basics of translation existed in the last universal common ancestor, many modifications separate, say, prokaryotes from eukaryotes. Now, if this is where the mRNA starts, and this is the start codon, which represents the first amino acid which will be added in the growing protein chain, Then here are a bunch of RNA nucleotides which will not become protein. They are referred to as what is known as the untranslated leader. So this is the leader, it's in the mRNA, but it comes prior to the start codon, not part of the protein. There are sequences here because proteins bind, and it's important to recognize where the start codon is, especially in prokaryotes, as I'll say in a second. And so some of the sequences in this untranslated leader may help to signal that this is actually the start codon, which is coming up next. Now, in our textbooks, we like to make pretty pictures and simplify things, saying AUG is the start codon. Once again, part of this is because cells have to be signaled, meaning starts here. AUG is, virtually always the start codon, but GUG and UUG are sometimes used in bacteria as well. And so given that there can be multiple AUG codons and possible other codons, these other sequences which are signaling a gene starts here, that is important. Also, just a message, methionine then serves two functions. So methionine is an amino acid, and the tRNA which binds methionine can bring it to where it is needed in a protein. So if you want to insert methionine in a specific part of the protein, you need this tRNA. But this is also then recognized as the start tRNA. So all proteins need, let's talk about eukaryotes first, need to start with this tRNA, which brings methionine. So all proteins in eukaryotes begin with methionine initially, although that can be removed later if that doesn't meet the structural needs of the protein. So in eukaryotes, this tRNA could either be the start methionine, and once again, that methionine could be removed later, or later in the protein could say a methionine belongs at this spot in the amino acid sequence. Prokaryotes, however, have two different forms of this tRNA. There's different tRNAs. So one form of the tRNA will bind to N-formal methionine. And so this is then different to start the protein than the regular old methionine, which will be inserted later. So in prokaryotes, you have methionine and N-formal methionine, a modification of it. So these two methionines are different, and these two tRNAs are different. This one can be used at the start sequence to start off a protein, whereas this one gets used if methionine is required later in the protein construction, but they are not interchangeable. So in eukaryotes, methionine and the tRNA for methionine are the same, whether they're used to begin a protein or not. But in prokaryotes, they are different. So with that introduction, let's then talk about the process of translation. First, it initiates, as I just described. And then if we were going to take the next step of elongation, we could break it into a couple of substeps. So first, the ribosome, once assembled, has two sites which will fit tRNAs. So there's what is called the P site, where the methionine bearing start tRNA has bound. But then next to it, there is an A site, which at the beginning was vacant, and that will now be filled by the tRNA whose anticodon here, these three nucleotides, matches the next codon in the RNA. So if the start codon was AUG, this first tRNA had the anticodon UAC, which was complementary to it. If this sequence is UUU, then the anticodon of the corresponding tRNA is AAA, and this would bind phenylalanine, for example, but that's not phenylalanine. The anticodon would then complement the codon. So we have two sites formed by the ribosome, a P site and an A site. Now, I'm not depicting them here, but there are proteins which are also helping with this. And once again, the tRNA, which can reside at this spot, has to have an anticodon which matches the codon of the mRNA. This is how the order of the nucleotides in the DNA copied into the RNA can then specify, put this amino acid here. Okay. Now, in step 2, a chemical reaction occurs, which arguably is one of the most important chemical reactions in life. Here we have two separate amino acids, but we would like to join them because at the product, we would like a chain of amino acids, a protein, where these are joined. Now, just a quick definition, amino acids get their name because they all have an amino group that's an NH2, the nitrogen, there is the pink one. So an NH2, and then a carboxylic acid group, C double bond O, OH. So each amino acid has an amino group and a carboxylic acid group, hence the name. And if you remember basic biochemistry, if this partner has a free OH, and that would be present on every amino acid. Meanwhile, each amino group has a free H. This then is opening the door for a dehydration reaction, where one partner gives an H, one partner gives an OH, and you've removed water, hence dehydration. But now these amino acids, they then miss their old partners, and so then they form a chemical covalent bond between the two, which in this case we will call a peptide bond. So we have two amino acids here, and I apologize, just in my animation, I'm not going to remove the OH and the H, I'm just going to smush them together. So it's not quite true to life. Sorry about that. And so we then smush them together, making a peptide bond. So in this ribosome, the amino acid in the P site is now going to be joined by a peptide bond to the amino acid in the A site. And we can refer to this enzymatic activity as what's called peptidyl transferase, if we wanted to give it a name. And we can say peptidyl transferase is the enzyme which then attaches these two so that they are now stuck together. Now, I'm older, and so that's how I was taught this process. We now know that it is not an enzyme, all right, peptidyl transferase, which is doing this, but actually the ribosomal RNA. So it is not a protein enzyme, but rather the ribosomal RNA acting as a ribozyme RNA, which could catalyze a chemical reaction. When I was in school, not only was it taught that that didn't occur, it was taught that that was impossible. And it was Nobel Prize winning work which demonstrated that RNA molecules were capable of catalyzing chemical reactions, that in addition to protein enzymes, there were RNA ribozymes. Not only is this possible that there are ribozymes, here performing one of the most essential tasks in cellular life, joining amino acids to make protein, it turns out that this is performed not by the proteins. Remember how many little proteins there are? When I was in school, it was just assumed that one of the proteins is doing this step. The proteins are holding the RNA together, and it's the RNA which does this step. So the ribosomal RNA acts as a ribozyme, forming the peptide bond between the amino acids in the A site and the P site. Once again, we can talk about the origin of life later. It is thought that life was first based on RNA before proteins became more and more involved. And the fact that here it is RNA in the ribosome, which is building the proteins, that helps to That's consistent with that idea. So once again, here's a different animation. So we have an amino acid in the P site. We have amino acid in the A site. Notice that all of them have a free H and a free OH. And if we then remove them, take the OH from one and the H from the amino group, then we've removed water. That's a dehydration reaction. These two atoms, they once had partners with whom they were sharing electrons. They no longer do, so they're looking to form new partners. And so here they then can, as the nitrogen of this amino group bonds to this carbon, forming a very stable bond, which we call a peptide bond. And so this tRNA, the first one that was in the P site, no longer has its amino acid attached. The amino acid is now attached to the amino acid in the A site, so it will float away. And as you'll see in the next animation, the tRNA from the A site is then becomes the new tRNA in the P site. And as we'll see, this process will continue. But the next time it happens, what's going to happen is that we have these two amino acids which will be attached to this one. So we will get more and more amino acids attached to each other as time goes on. We start with the first one being attached to the second one, but then later it's an entire chain which gets attached to the next tRNA and its amino acid. And so the third step in the elongation process is that the ribosome will move. First, the large subunit moves, and then the small subunit moves. When it does so, this tRNA that was in the P site is no longer in the P site, and it will float away. In prokaryotes, there is a temporary E site or exit site that it could occupy. No such site exists in the eukaryotic ribosome. So this tRNA is done. It needs to then go and find that enzyme, the synthetase, which will attach a new methionine to this end of the amino acid, and then it could then be thus charged again and then used again. So these tRNAs will then be recycled as new amino acids get added. This blue tRNA was in the A site, but now it is in the P site. Notice that it now has two amino acids attached to it. The methionine that had been attached to the first tRNA, and now the second one looks like lysine. No, it's not lysine. I forget which one that is. I'm sorry. The A site is now vacant. And a new tRNA will then be brought here, whose anticodon matches the third codon. So this anticodon matched the first mRNA codon. This anticodon matched the second one. And now here's the third mRNA codon. A new tRNA will then bring a new tRNA will bring an amino acid to this spot, but only the tRNA which matches that third mRNA codon. Okay, and so the chain elongates, and then the process will just continue where a new tRNA comes in. The amino acids are attached together. The RNA acts as peptidyl transferase, a ribozyme, which catalyzes a chemical reaction, and so then the chain gets longer. So now, instead of two amino acids, there are three amino acids which are attached. Then another tRNA will come in which matches the next mRNA codon, and so forth. And now we have 4 amino acids. So these steps will simply then repeat, that a tRNA fills the vacant A site, the RNA acts as a ribozyme attaching these chain of amino acids to the new one, and then the ribosome then moves so that the now empty tRNA floats away, and the A site is now vacant again. And so by the end of this process, we have a chain of amino acids. This then continues until the ribosome reaches what's known as a stop codon. This stop codon will then end translation. There are 3 of these: UAA, UAG, and UGA. There is no tRNA which corresponds with those sequences. So once we get there, all right, so here is a tRNA whose anticodon matches this codon. But if this next codon is UGA, there is no tRNA which matches it. So the process will now stop, and the chain of amino acids is now finished as a protein. Now, when this happens, the subunits of the ribosome, they will then disassociate, and all of these will then go their separate ways. Now, as you might have guessed, there are proteins which I have omitted from this process. So there are a couple of other factors which will then be integral here. But the important point is once we get to this point where those other proteins have bound and there is no tRNA because the mRNA codon was the stop codon, then the process will stop. The chain of amino acids will be separated from the last tRNA by enzymes. This is now a protein, the final product of translation. The tRNA will then be charged again, and the ribosomal subunits will dissociate. Now, if this is the stop codon, notice that the mRNA isn't done. There's more nucleotides here, but they will not become protein. There is an untranslated trailer. So when you look at an mRNA, the first part does not become protein. That is the untranslated leader up until the start codon. And then after the stop codon, that part does not become protein. That is the untranslated trailer. So this then is the process of translation. Now, it took me a while to talk about that, but these steps where amino acids are being added, that can happen, say, 12 to 17 times per second in prokaryotes. So 12 times in a second, new amino acids are being added to a growing chain. In eukaryotes, it's about twice a second. Now, there is far more to say, but that is perhaps more appropriately mentioned in the topic of the regulation of genes, which comes later. So once a protein is made, it then has more things to go through, like it has to be folded correctly. It may have to be modified in the Golgi. And so, this protein is not yet functional, but talking about that would then involve other steps. And then also, I would like to just mention that these next couple topics exist, but also delve into those more when it comes to the regulation of genes. So I had mentioned that there were all of these proteins which can be involved in ribosomes. But one of the fascinating things that we're learning is that they can vary, and they can have roles then in the regulation of genes. And so while most proteins seem to be present in all ribosomes, there are differences. So not all ribosomes then are equal. Some have proteins that the others don't. And thus then some ribosomes have unique roles. And so when we talk about the regulation of genes, one of the ways that we can regulate genes is by changing the proteins involved in the ribosomes. So for example, during embryonic development, if you wanted to translate the HOX genes, that is apparently preferentially done with ribosomes which have different proteins than a regular old ribosome, which is translating other proteins. So that is fascinating. Once again, perhaps a topic for later in gene regulation. All right, and so one of the things, once again, a topic that we're beginning to understand more about is the proteins in the ribosome, but then there can be what are called ribosome-associated proteins, RAPs. And so the making of protein is obviously a fundamental aspect of life. And so there are lots of other factors involved. A point I've made before, and so here it's saying that, when we go through different parts of the cell cycle, we can then make proteins associated with ribosomes, which can then, change levels of translation at different parts of the cell cycle. One important point then is, let's imagine that this is the genome. This is all of the DNA of the cell, which obviously in humans is made on the 2 sets of 23 chromosomes. Well, much, but not all of it gets transcribed, maybe 50 to 70%. It then forms what's called the transcriptome, all right, so the DNA which is converted into RNA. And then some of that then becomes what's called the proteome, the part which is actually converted into proteins which are performing functions in the cell. But if you ask what percentage of the genome is actually then translated into amino acid sequences of proteins, it's really only about 3%. All right, so the proteome is by far the minority of the genome. And so not only is it important to make proteins with the right amino acid, but it's important to regulate that so the proteins are made appropriately. So if you ask, well, what is the rest of this doing? It's not coding for protein. Much of it is regulating when the proteins are made. So that's a very important topic for the future. And then also notice that I said that there were tRNAs, that there were rRNAs, and there's more. There are a lot of RNAs which are never converted to protein. So while translation, conversion of the DNA message into an RNA message into the amino acid order of a protein, that's important. A lot of what runs a cell is actually RNA, which is never converted into a protein. Just important to mention. So sometimes in biology, we use the term the central dogma, the idea that the genetic code of cells is DNA, which replicates itself, that then can be transcribed into RNA, messenger RNA, and the messenger RNA can then be translated into the amino acid order of a protein. That's what this topic was about today. And when we look in the mirror, we see, the keratin of our skin or hair. Oh, well, that's protein that was made through translation. The opsins in our eye, which are perceiving light, well, that's protein. The dopamine receptors, which let us think, or the taste or smell receptors, which let us perceive the world, that those are proteins. The hemoglobin in our blood carrying oxygen, the antibodies in our blood fighting microbes, the digestive enzymes in our digestive system, all of these are proteins. So to a large degree, proteins run the cell and run the organism. And this process of translation is then thought to be, and not only critical for all of us today, but one of the essential building blocks of modern cellular life.